where we're going to be talking with Steve Quinn, Dr. Steve Quinn at the University of York, and Hermia Kung, who is absolutely brilliant as a project student, a summer undergraduate project student, using an EI Flex at the Centre of Excellence in York, which we established just over the summer last year. So more about that in a minute. I also wanted just to take this opportunity to give you all a quick update on where we're at as a company. There's a lot of exciting things going on. It's in the name. We have to lean into the puns. I'm afraid it's the only way to do it. Otherwise, we all go mad. What are we about? I think most of you know it's about single molecule spectroscopy simplified. So really making single molecule measurements really easy for everybody. It was founded out of my lab here in Sheffield between myself and my co-founder Rob Bell, who brought all the software and the actual business acumen. We've both learned both things together. So he is now really excellent on all of the single molecule stuff, and I am getting there on some of the business stuff. So it's good. As you all know, that new tools is really what drives new discoveries, and that's no different in our business to telescopes or microscopes or anything else. And so our instrumentation does enable that direct single molecule detection, so detecting those individual molecules, the shapes they're in by single molecule FRET, and how they interact either by FRET or by FCS and FCCS. The data that, the raw data kind of look a bit like this. You'll have seen these things before. We've got different conformations of our molecules. Most of the time, nothing in the volume, and occasionally you see a burst of fluorescence. That's a molecule in the volume. Look, we weren't the first people to do this. Other people have done it, but it's a lot more expensive. It's much more complicated and slow. And so really what we came up with is this benchtop connected device, which looks like this. It's 2 foot by 2 foot and critically sits on the optical, doesn't sit on an optical table, right? That's absolutely essential. Just sits on your benchtop, right? We installed one in Copenhagen a couple of months back, right next to floor to ceiling windows, sunlight streaming in, absolutely fine, right? So you can use it under you don't need any laser safety. It's a class 1 laser product, and it works under ambient light conditions, which I think is really, really critical. And we boxed all of the difficult stuff into the software to make it really easy to use. And I think Hermia is going to really speak to some of that in a moment. We've been really supported by our first early pioneers, and Steve actually isn't on this one, and he gets his own special mention in a minute. So in York, but this is us here in Sheffield. We've got things in Warwick, we've got things across the US, and we've actually now got 12 instruments out in the wild, with about another 12 to go in over the next 6 months or so. So it's all flying off the shelves, which is super exciting, and people are doing some great science with it. The people are really seeing the effects of these, of how easy it is to use, big demand, and making things possible that really wasn't possible before. Right, enough from me. I'm going to hand over to Steve now, who's going to just give you a bit of an intro to himself and also to the Centre of Excellence at York. After that, we'll have a very short discussion around why the Flex was the right instrument for him, and then we'll move into Hermia and some actual science and data on her topic. So, Steve, over to you.
Well, thanks very much, Tim, and good evening to those of you who are in the UK. Good morning to those of you in the US. So I have a really, really unhealthy interest in Alzheimer's disease and more recently virology. And we are primarily a single molecule fluorescence lab. So we have ERF instrumentation, we have time correlated single photon counting instrumentation, and as Tim has just mentioned, more recently we have introduced the EI Flex system into the University of York. So we use this series of single molecule tools to really explore a variety of protein interactions, protein DNA interactions. More recently, we have looked at protein vesicle interactions, and we are very, very open to collaboration. So as Tim said, we have the EI Flex as part of the Exciting Instruments Centre of Excellence at the University of York. That's located in our so-called technology facility. So this is a facility overseen by Dr. Peter O'Toole, and it basically encompasses a variety of equipment and infrastructure, including the EI Flex, we are very, very open for those of you who want to screen your samples or perhaps generate preliminary data, or perhaps even come on down to York to operate and learn how to use the instrument yourself. Please do get in touch if you would like to do so. So all in all, we're a single molecule biophysics lab. We explore protein-protein, protein-DNA, DNA, RNA interactions, more recently vesicle protein interactions. And you, as Tim suggested, are going to hear a little bit more about some of the, I hope, exciting, no pun intended, data that Hermia has collected over just a short period of time last summer.
Brilliant. And just before we get to that, Steve, it'd just be good to understand, what was your decision-making process that you're going through, that made you think that an EI Flex was going to be a good instrument for you in your situation?
Yeah, I mean, so there's a couple of points here. I mean, the wide range applicability of the system to not just protein-protein interactions or protein-DNA interactions, but to a wide range of biological problems was I think first and foremost the underlying reason why we decided to bring an EI Flex into York. There are some technical reasons as well. I think having alternating laser excitation gives us an ability to measure the so-called particle stoichiometry that tells us how efficiently our molecule is or is not doubly labeled. That has been a game changer in trying to identify and label quite complicated systems, especially RNA constructs. The EI Flex capabilities are not limited to single molecule FRET detection, but as you mentioned, Tim, you also have the ability to perform FCS and FCCS. So you can use the same instrument to extract parameters like particle size. I mean, this morning, for example, in the 30 minutes that I had spare between meetings, I quite literally ran into the lab and collected data on lipid vesicles undergoing, conformational changes in response to proteins, and got that information and that data out super quick. So having an ability to generate data really, really quickly and get informative insights, whilst not needing a dark optical room, has also been quite important for us.
Brilliant.
The system that we have in York has temperature control. Hermia is going to show you some really beautiful data that she and others have collected, by ramping up the temperature on DNA molecules. But it also gives us an opportunity to hold proteins at specific temperatures. So again, that's been super useful. And again, no pun intended, or maybe I should say pun intended, but the excitation and the emission lines have good flexibility, right? So we can switch between blue or green excitation or red excitation. So we're not limited to a choice of or a specific donor and acceptor FRET pair or a specific fluorophore for FCS. There's quite a lot of flexibility there.
Brilliant.
We got
And so,
data.
Steve, tell us a bit about just, the number of, have you got a lot of uptake of this instrument? How's the usage going? What's the, what's been the response when
Yeah.
from when you put it in?
So we can you believe it, Tim? It's been about a year, I think, since we put the Flex in. Genuinely, and this isn't a sales pitch at all. Genuinely, it's been used from physicists to biochemists to biomedical engineers at our university. It's been used by structural biologists, and the instrument, since being installed in March, has genuinely been used on a daily basis from not just 9 till 5, but throughout all hours of the day. I think this has really been a game changer for not just my own research, but for the research of numerous research labs around the university, and it's encouraged collaboration. And it's encouraged grant writing, and it has allowed us to secure, additional funding, because by leveraging single molecule FRET, which I think is, genuinely at the forefront of modern science, you can answer some really important biomedical and photophysical questions. So yeah, it's been used by a wide range of people, a wide range of groups, everyone from undergraduate project students to professors and PIs and postdocs, all the way through to summer students like Hermia that you're going to hear from soon.
Brilliant. That's great, Steve. Well, without further ado, I think everyone's itching to get to the science, and they're like, "Oh, come on, Tim, let's hear the science." So Hermia, if you'd like to share your screen, that would be great. Hermia was selected from a very strong pool, I've got to say, of applicants for the summer student that we kind of sponsored as part of the setting up of as York as a centre of excellence, and we were really super impressed, I've got to say, with the amount of the her work ethic, but the sort of the data that she got. So without further ado, Hermia, over to you.
Hi, so I'm Hermia. So I'm going to be talking about the project I did in the summer and titled Unravelling the Flexibility of DNA and RNA Systems. So I'm a current third year natural scientist at the University of York. And so in my second year, I took a 10-week generation research studentship sponsored by Exciting Instruments. I was supervised by Tim, Steve, and Dr. Peter O'Toole to quantify the conformational changes of DNA hairpins and mRNA elements using the EI-Flex, which is now the Exciting Instruments Centre of Excellence at York. And so you can see here me with Steve and Pete, me with the microscope, and then you can test your samples as well at the University of York. I was super lucky to spend 10 weeks doing 2 projects. So I'll talk about my first project first, which was using the DNA hairpins. So this is the hairpin we used. You can see that we tested different length loops of hairpins. So we had 31A, so adenosine bases, 40A and 50A hairpins, and we changed the salt concentrations and the temperature to see whether the environment would change the open or closed favorability of the hairpins. So we attached dyes, so the Cy3 donor dye and the Cy5 acceptor dyes. And so you can see that in the open hairpin, they are much further apart, and then in the closed hairpin, they are much closer together. And that's what the single molecule FRET measurements can quantify. So we use the fluorescent tagging and single molecule FRET to monitor these unfolding kinetics. So how does this happen? So we have single molecules floating through the confocal lens, as you can see here. And then depending on whether it's open or closed, they'll have different FRET efficiencies and different sized bursts will be seen. So we use the exciting, they become excited using the ALEX, so alternating excitation layers, so green and red alternating. And the efficiency is determined by the ratio of acceptor emissions to the sum of the donor and acceptor emissions due to donor excitation. So here the blue, the blue one is the donor excitations, and the acceptor emissions due to the donor excitations, and then the red is the alpha emissions, so the acceptor emissions due to acceptor excitations. So you find the ratio of those to determine the FRET efficiency, and if they are further apart in the open conformation, then the FRET efficiency would be lower compared to the closed conformation. So here you can see what the histograms look like after the single molecule FRET experiment. And on the left, you have the all-channel burst search, so unfiltered bursts. And we can distinguish out 3 populations. So we have the donor only at the stoichiometry of 1, the acceptor only at stoichiometry of 0. So this means that the DNA hairpins are only singly labeled with either donor only dyes or acceptor only dyes, but we're only interested in the middle at stoichiometry of 0.5. And this tells us that the hairpins are doubly labeled. And these are the hairpins that we're interested in. So first, we looked at salt concentration. So I increased the salts from 0 to 100 millimoles of salt. And you can clearly see 2 separate populations in the histograms, one for the closed hairpin and one for the open hairpin. So we have a high FRET state and a low FRET state. So the high fret is when the hairpins are closed and the dyes are really close together, and there's the energy transfer from the donor to the acceptor dye is really high in efficiency, around 0.7. And then in the open hairpin, as the dyes are further away, we get the low fret population around 0.1. So you can see that increasing the salt concentrations, you can see the high fret population growing and then the low fret population decreasing as well. So this, I was able to replicate Sarah's PhD student's results from before in my first week of my summer studentship. And you can see that there's a clear trend and all of the graphs were reproducible as well. So after this, I then chose 40 millimoles of salt where there's a good population of both high fret and low fret to investigate how temperature plays a role in opening and closing these hairpins. So you can see here, so this is a 31A loop length of DNA hairpins, and I increase the temperatures from 25 degrees to 45 degrees. And you can see the low fret population begins to grow again. So high temperature favors the open hairpins. And this aligns with that hypothesis that it's the 6. So if we go back quickly to here, it's these purple 6 base nucleotides that separate when the hairpin opens. So adding heat will provide the energy to break these bonds, and so high temperatures would open the hairpins. And so if we go back to our temperature ones, we can see that that confirms our hypotheses. So we use single molecule frets of 31A DNA hairpins at fixed 40 millimoles of salt and varying temperatures. But how can we quantify this change and this growth of the low population state? We can find the equilibrium constant for the closing reaction from dividing the integral of the closed hairpins, so the area underneath the high fret population, by the area underneath the low fret population. And this is useful because we can convert this into the change in Gibbs free energy at each temperature. And so we can then quantify how the favorability increases of the open hairpins as the temperature increases. So we repeated this for the different length loops, so 40A and the 50A hairpins, to produce this graph of change in Gibbs free energy against temperature. So this is useful because you can see that if the Gibbs free energy, the change in Gibbs free energy is above 0, that means that the open conformation is, or the closed conformation is less favored. And then if it's less than 0, then closing the hairpins is much more favorable. So we can see here that we have the 50A at the top, the 40A and the 31A. So the longest loop at the top all the way down to 31A. And we can, from this graph, we can extrapolate the change in enthalpy and the change in entropy values. So the change in enthalpy from the gradient and the change in entropy from the intercept. What this tells us is that high temperatures destabilize the hairpin, leading to opening. Longer loop hairpins have higher delta G values. So at every single temperature, the blue 50A loop has a greater value than all the other 40A and 31A lengths. This means that closing the hairpin is less favored for longer loops. And this is kind of what we thought would happen before we did these experiments, because if it's a longer length loop, then it can take up has more degrees of freedom in its open state, because it can like move around a lot more compared to when it's shut. So it'll be harder to shut them. And finally, there's variation in both the change in enthalpy and the change in entropy. So we expected for the change in enthalpy to be constant for all of the length loops because it was the same 6 nucleotide sequence, the purple sequence, that would be breaking to open the hairpins. And so the length of the loop shouldn't, well, we didn't think would disrupt that or have any effect on that. But because there is changes and variation, it indicates that stacking interactions occur within the loop itself. So the loop may rearrange itself into a helix or just stack along each other. And there's more stacking that can happen if the loop is longer. So that's the results I got for the DNA hairpins. We'll move on to what I did for mRNA. So I studied the TMEV mRNA, which is a virus. And viruses hijack its host ribosomes during viral protein synthesis. So what we wanted to investigate was programmed ribosomal frame shifting. So viral gene expression is regulated by programmed ribosomal frame shifting, where the ribosome slips into an alternative open reading frame. So you can see down here that shifting back one frame, that the codons that are read are completely different, are using the same length of genetic code. So this allows for higher protein diversity within the viruses, and it's often used to control the ratio of enzymatic and structural proteins in viruses. For TMEV in particular, it requires the protein 2A binding to the mRNA to stimulate the ribosomal frame shifting. However, this mechanism is highly unknown. So I set out to try to investigate the mechanism more with single molecule FRET. So how we did this was we put on the dyes on the mRNA. So you can see the Cy5 donor and the Cy3 receptor dyes. And in its unbound state, so when it's not bound to 2A, the protein, the dyes are around 29.4 angstroms apart. Whereas in the pseudoknots, they are stretched further apart. So it is hypothesized that when 2A binds, the G and the C, so the arrows here, will kind of bind to each other and then form the pseudoknot you see on the right. And in the pseudoknots, the dyes are further away, which would give rise to a lower FRET state. So I've got these labels modeled up. So I'm sorry about that, but the pseudoknot should be the lower FRET state, and then the unbound state, when the dyes are closer together, will be the high FRET state. So you can see here that at when there's no 2A protein present, and all of the mRNA is in this unbound state, there's only one high FRET peak. Whereas as I add more and more protein 2A to the mRNA, you see this growth of this lower FRET population, which we think is the pseudoknot and aligns with what we predict would happen. So it was really interesting to be able to work with both DNA and mRNA using the EI-Flex. And you can see that I was able to gain some really nice graphs from this as well. So overall, in only 10 weeks, I was able to be trained on the EI-Flex, and I analyzed its data using Python. I carried out nearly 100 single molecule FRET experiments that were reproducible, reliable, and informative. And I was able to do that due to how easy it was to use the EI-Flex, how user-friendly it was, and the mentoring I got at University of York. And then I identified that simple DNA hairpins are regulated by environmental factors such as salt and temperature and behavior dependent on loop length. And new data reviews that conformational switching of TMEV RNA in response to protein 2A, which may have antiviral implications. So thank you for listening. And if you want to test your samples at the University of York, you can contact Steve and Pete, and the emails are found below there. Great.
Really great stuff, Amir. I'm really keen to throw that open to the floor for any questions from the audience. If you want to just either raise your hand or type questions in the chat, you'd be very welcome. In the meantime, I've got a couple, I guess, and but while people get comfortable, I think you touched on this, but it was counterintuitive that the enthalpy was the thing that showed the biggest change rather than the entropy for your loop length experiments. What was the thinking behind why that was?
So we thought that, yeah, it was the loop length itself of the adenosine bases that was interacting with itself. And when it interacts, it will stack. And then so it would require like more energy as well to break those interactions to open the hairpin further, which is why there is a variation in the enthalpy, whereas we thought that the length loop would not affect the change in enthalpy of opening the hairpins at all.
Interesting. And did you do any experiments to get out look at the activation energies of that? Because I guess you could get kinetics from these experiments as well. Did you do any of that analysis, or is that something for the future?
I didn't do any of the analysis for the activation, but I think we did speak about it at the end, like right at the end of my 10 weeks. And Steve was like, yeah, maybe a future student could do some more like FCS analysis on it, I think, Steve.
So I think we can do it literally on the data that you've got.
Oh yeah.
We've got, I think Paul Harris is on the call here on the meeting here. He's developed a really nice H2MM type approaches, photon by photon.
Yeah.
There's the Klaus Seidel and others have developed the dynamic. Oh, hi Paul. Have developed the sort of dynamic PDA, photon distribution analysis, that we've used, especially for these hairpin dynamics, and then that might just give you another level of detail than just the equilibrium measurements, which are still obviously really useful. I mean, it's interesting. I think we don't see many temperature studies at single molecule level, right? And I think that's actually quite interesting to be able to pick out. And one of the reasons why it'd be good to do that analysis, and actually even just on the current data that you've already got, we could run that through, so there's stuff to talk about there. Any questions from the audience? Just feel free to click the the react button and raise your hand, or ask any questions that you've got. Anything from anyone there? Otherwise, I'm just going to keep going. So that's your fault, guys. You've said a bit, Hermia, but it'd be good to get a feel for this. And can you just tell us a bit about the situation? Because I think you've been there about a week, right? And we rocked up with one of our investors who was trying to work out whether they wanted to actually give us money if the instrument worked or not. We kind of put you on the spot. Do you want to just explain what happened in that meeting and what we made you do?
I mean, yeah, it was like 1 week in. It was my first time meeting Tim and Mahmood and everyone from EI Flex, I think. And then you were like, oh, is it okay if we bring in an investor and you can just talk around like how to use the EI Flex? And I thought, well, I've only been here a week. I've used the machine like only a handful of times. I'm not sure if I can, but because it was so easy to use and all the functions were quite self-explanatory as well, I was able to do, I think, a good explanation of how to use the EI Flex. And I'm really glad that I think you guys appreciated my efforts in trying to explain it. I was very nervous for it.
And I mean, look, I can only apologize. We massively put you on the spot, but it got us the money. So the guy was super impressed, and as were we, especially at the end. As afterwards, we said, "Oh, she's only a second year," and he was expecting us to say PhD student, not undergraduate, right? So look again in the comments, Akash, thanks for that. Just saying really great presentation and really clear. Nicole's question, I'll just read out to you. So lovely work, Hermia. Did you notice that your data became messier at higher temperatures? Wondering about any effect of temperature on the dyes.
We did do some control experiments on the dyes. So we used the photometer, I think it was, at different temperatures, but the profiles that we saw, that the dyes were not affected at all by the temperature. And we did loads of repeat experiments as well. And how the dyes emitted light and the fluorescence of it was unchanged with temperature. So the only reason it could have been messier was just because potentially if there's less of like a low fret state being detected or less of a high fret state being detected, it's hard to fit the Gaussians or fit the data better, which might give rise to the messier kind of look.
No, that's fair. And I think when you get to the kinetics as well, something we've noticed is that when you move, when you get to the extremes, you get fewer events where you're actually converting between the two states, so the rates become harder. Go on, Steve. You just butt in.
One thing that was that's really great, and just in response to Nicole's comment, is what we can do with the flex is we can also not just do single molecule FRET, we can do a burst analysis. So you can look at effectively the brightness of single molecules as a function of your experimental condition. And certainly one thing that we're really interested in is to explore the photophysical properties of, not just cyanine dyes, but other fluorophores using the flex. In the context of Hermia's work, there wasn't too much variability as a function of temperature, which was great. Similar story with sodium chloride, not too much variability. We have seen interesting effects recently, though, when we add ATP into fluorescent dyes. So we don't quite have the answers there, but the EI flex has certainly been super useful to not just quantify FRET, but photophysics.
Brilliant. Did you want to come back on any of that, Nicole?
No, thanks. I was just writing thank you in the chat. But, thank you. Great answer.
Nice to see you. Great. So, someone else has written amazing work. We're just wondering if there's a possibility of measuring FRET in RNA condensates. I'm happy to take that one. Absolutely. In fact, the V2 that we're bringing out shortly will have XY capability, so you can position yourself to do experiments both in and out of condensates. So that will be actually quite helpful, I think, because I think for a lot of people there's questions around mechanism of action, and you want to compare the dilute phase to the condensate phase, so you need to be able to know which one you're in, and that's something that's clearly interesting and absolutely possible. To come to Aitan's question. Hi, Aitan. Thanks for joining. Aitan's asking, as for the entropy-enthalpy effects in your hairpin experiments, what do you think the results will be if you will test poly T loops rather than poly A?
Aitan, maybe I can jump in. We collected that data and we've looked at it literally this morning. We don't have the answers yet, but we have all that data ready to be processed. So we're hoping to address that question.
So put you on the spot then, Steve. So what's the hypothesis? I mean, RTs. And Aitan, maybe you can jump in here because you know a lot about this stuff. Is there a different? Obviously, the base pair stackings have been well looked at, right? But,
I mean, it's all about the flexibility of the polymer, and in enthalpy terms, I guess it's about the competition between the annealing of the stem and other intramolecular interactions within the single-stranded DNAs. So I would assume that you would still have enthalpy and entropy dependencies, but that the enthalpy dependencies will be much less pronounced.
Yep, and that's effectively what we see. But watch this space for the data to emerge. By the way, all of this work, or all of the hairpin work, wouldn't be possible without Roman Sukhanov's input. So I see Roman on the call. Thanks so much for your help, Roman, and Ayel near. So if Ayel's on the call as well, thanks for all of your hard work on the early days in this.
Brilliant. So there's another question here. Excellent talk. Has the system been tested for membrane proteins? I can take that one. We're doing yes is the answer. So you can work with membrane proteins in a number of ways. One of those is in detergent solubilized vesicle type modes. The other thing is in the way that we work with them in SMALP, so cookie cuttering them out of the thing. Steve actually works with whole vesicles. Go on, Steve.
Yeah, but also model membrane nanodiscs. So we've incorporated membrane receptors into membrane nanodiscs and then looked at conformational changes as these nanodiscs containing single receptors diffuse through the confocal volume. So yep, entirely possible on those systems, but also on lipid vesicles.
Yeah, and the other bit of work we've been doing here is from on the FCS side, and when we're looking at complex complexes, so not just a single GPCR, but actually GPCR in complex with a ramp, for example, ramp protein that then makes a particular receptor. Actually, we've got some really great data. A PhD student in my lab, where I do still spend some of my time occasionally, on the research side, Georgia's got some great data showing how she's using FCS to prove that she's got both the GPCR and the ramp in the same nanodisc, right, or the SMALP, either way, but in the same exact, together, and that, diffusing together, so that she can then use that as a screen, which we're also doing in a 96-well plate format, which I'm not supposed to tell you that because we've got a high throughput instrument, which I'm not allowed to talk about. But anyway, we have a high throughput instrument, which I'm not allowed to talk about, which will be finished and ready for the wider world, later this year. So, but yeah, that gives us a lot of opportunity for drug screening and capacity there. So yeah, great. Any other questions, comments, thoughts? Brilliant. In which case, I'd like to just close out by saying thank you to everyone again for coming. I know that this is a slightly shorter notice than we've had before. We've been, can I say, a little busy, I guess. A lot of us were at Biophysical Society meeting. It's good to see a lot of friendly faces there, from that. And I think that the key next steps, Steve, it'd be good, what are the next steps, right? What are the challenges for the single molecule FRET field? And I'd throw this open, we've got a lot of people on the call who, who are really active in this space. What are the next big things for this space? Steve, do you want to just start there?
Yeah, I mean, in terms of my my own sort of research group, I mean, there's clearly technical questions and biological questions that we want to address. One of the big challenges that from an Alzheimer's disease perspective we face is how are these proteins, which are neurotoxic in the brain, how do they disrupt biological membranes? So the EIFlex has been really helpful for us to try and address specific questions. High level questions, wouldn't it be great to do some single molecule analysis inside an animal at some point? I know that folks are working on tracking quantum dots inside mice and rats these days. Wouldn't it be great to see some single molecule spectroscopy inside a real living system? I think there's lots to be excited about in the community. There's so much work going on, from DNA protein interactions to protein self-assembly to RNA work, as you've just heard, with antiviral implications or implications. I think genuinely it's a great time to be a part of this community, and thanks to you, Tim, for building this community here in the UK and beyond.
Brilliant. Any again, open to the floor. What are the next big challenges, right? What would you like us to be focusing on? Maybe I can help a little bit there. I think for us as a company, what we're looking to do is make this as easy as possible, right? I guess that's clear from everything we've talked about. We feel like that we've solved part of the problem by providing a turnkey instrument, but that's only part of the solution, because now, we're looking at, well, what's the next steps, right? And so, we've solved that bit. So now it's data analysis. A lot of that is getting there. We're in the cloud. We've got FretBurst with, that Paul Harris has been massively supporting. Eitan Lerner has been growing a lot of the analysis piece there with the H2MM side of things as well. So for our side, we're actually now, we've just hired a stellar software engineer who's we've basically in 2 weeks given him a job to actually generate an entirely bespoke piece of desktop analysis software that's just going to do everything in one place for people who don't really want to engage with Python or MATLAB, which we feel you, we understand that, we feel your pain there. So we are solving that piece. I think for us, the next piece is actually the other end in terms of reagent generation. And so it'd be interesting, you know, this is a topic for a broader discussion, but, you know, what are the challenges there, right, in terms of label protein, in terms of where you put the dyes? We're putting together a piece of software that will help you to decide where to label, you know, your proteins if you're trying to capture a particular conformational change, or if maybe even you just don't know, right, what's going on. You know, something that will walk you through where to put your dyes and your best labeling strategy. But then we're also working on a bunch of things around actually just providing you with a kit for cell-free labeling expression, and cracking on with, you know, because the beauty of single molecule, let's not forget, it is one molecule at a time, but you also need absolutely sod all material, right? So actually you can do this, you can generate from cell-free expression very small amounts of material, which is perfect for what we need, which is great. Good. We've had a couple of other comments come through. So Ji Yang has said, beautiful data. Have you calculated the if the distances from FRET are consistent with the structure of the mRNA? I wonder how small changes of distance flex can detect from FRET efficiencies. So Steve, have you done the accurate FRET corrections, or not worried about that bit yet?
So yes, we have. They are consistent, and watch this space for a bioRxiv paper coming to your screens soon. Brilliant.
So that's great. And just on in terms of the amount of distance that we can detect, we know that we've done this on a DLA polymerase systems, where we can detect you know, sort of sub 2 angstrom changes in distance, where you get a very, you know, especially if you're around the 0.5 FRET efficiency, which is where you're most sensitive to distance changes. But you can absolutely titrate in. So we did some experiments where, for example, you've got an open, a closed, but you've also, when you put an incorrect nucleotide, you get a partially closed state. And literally that is less than a 2 angstrom change. And you can titrate that absolutely reproducibly and see a sub 2 angstrom change in the structure. So that's great. Roman's asking, yeah, pleasure, thanks, Shi. Roman is asking if the flex is compatible with fluorescence lifetimes. It's compatible with, but it doesn't do them yet. So if you really that's a slightly facetious answer. Yes, if you want pulse lasers and pulse electronics, that's absolutely possible with the hardware in place at the moment, but it's not an option that we currently offer. What we from the work that we published a while back in Nature Methods in 2018, and the follow-up work even on the proteins in 2023-24, we're headed by Torben Cordes, the last one, and Torsten Fugel, the first. Actually, you get you know, 90% of the use cases are actually better served by the intensity-based measurements. But if you want the lifetimes, talk to us, because, you know, that's something that we're happy to discuss for individual use cases if that becomes particularly important. So yeah, exactly as Aitan said, you need an Alex version just with pulse lasers and the TCSV electronics. And as Aitan points out, you know, as he said, this will come with the expense of the photon rate. So we had this discussion at BPS with Jelle Hendricks and others, and we went back to the very first Pi paper and Alex paper, which was great actually. The reality is, when you pulse, the very nature of the fact is you send your pulse and then you wait maybe 12.5 or 20 nanoseconds, 25 nanoseconds, depending on exactly how you're doing it and your rep rate of your lasers, before you pulse again. And what that means is, and actually we have done that side-by-side comparison for our instrument versus others that use the pulse lasers, we actually, it's quite remarkable. Under the same conditions on the normal working, we seem to be getting around 10 times the number of photons, which broadly speaking is not unreasonable. If you've got a 2 nanosecond lifetime, but you're waiting 25 nanoseconds between pulses, that's kind of why, right? Because our lasers are always on, you're getting every photon you possibly can. There is another advantage for our system, that you're not driving things to the triplet, because that pulse is super high power for the pulse when it happens, right? And so actually there is a danger that you excite things into higher states and actually drive things into the triplet, which has been there. So yeah, Eitan, go on, just come on and ask your tech question. No, you're muted. You can't ask a tech question with liquid.
Yeah. My bad. So, but you can still incorporate the TCSPC card and perhaps think about having nanosecond FCS on top of the alternating laser excitation.
Yes,
as long as the alternation is slow enough.
Those two things are slightly different. So nanosecond FCS requires continuous wave lasers,
which you have,
that are super stable. And absolutely, V2, we are looking to support that, because I think there's some really interesting applications in terms of overall in IDP space, in overall chain configurability, conformational dynamics, and general. That piece definitely we're really interested in and on. And absolutely the TCSPC piece is doable. It's just more expensive, as you say, as well, not just in terms of photon budget, but in terms of budget. So it's absolutely doable. It depends a little bit on individual customer need, but we are looking at building that out, and that's one of the beauties of the situation that we're in now, with the ability to invest in the expansions to the instrumentation offering. I still hold that 90% of the experiments that most people will want to do. Now, that isn't to say it's not useful to go and do a time resolved measurement. It is, but do you need that data in every single one of your measurements? No. No, that's the reality. So you might want to go and do a control experiment somewhere, and then having and that could be an ensemble TCSP measurement where you're just you're looking at, for example, the key one is how freely are my dyes rotating, right? So actually going out and doing that experiment, perfect, absolutely, but yeah, for people who really want it, in Steve's words, watch this space. Fantastic. Any other questions from the room or anywhere else? I'm not sure where else that would be, in fairness, but Awesome. In which case, I would very much like to thank, especially Hermia. That was a fantastic presentation. I think it shows your own ability. You see the rounds of applause coming through. And of course the ease of use of the instrument, of course. But you made it work, right? You worked super hard. You got great data, and I think it's going to have contributed to a number of papers, right? I think it's not just one, right? So that's pretty good going for 10 weeks of an undergraduate project. Of course, if anyone's keen to reach out to us around any of the things that have happened here, Steve and Hermia and Peter O'Toole are super happy to host people and to talk to you about taking data there. If there's data or preliminary data you would want to get a flex in your lab and you need preliminary data, that's an option. You can also talk directly to us. We'd love to show you our new premises in a couple of months' time, so you'd be very welcome to come and and get data there as well. But talk to Steve too, because sometimes the unvarnished version from an actual user is that much more sort of reliable than anything you'll get from me. But yeah, fantastic. And with that, we look forward to the next one. We're going to be starting to ramp up the regularity of these, and so we imagine we're going to be putting one of these out a month. So we look forward to seeing you all soon. Thanks for joining us, and we look forward to the next steps. See you soon, guys.